about the question number 5. Define and design a test cross. So first we have to define a test cross, then we will going to design test cross. So test cross is just an experiment in which organism showing dominance for a trait has to be tested for its genotype. This is the basic definition of a test cross, that test cross is an experiment. Test cross is an experiment showing a dominance, in which organism is showing dominance for a trait has to be tested for its genotype. To test the genotype of an organism, we use recessive allele, recessive pair of alleles. Alright. Now, when an organism is showing dominance for a trait, it could be homozygous in condition and it could be heterozygous in condition. Now, when we experiment, when we do experiment to examine the genotype of an organism, we know that we use recessive pair of allele, recessive parent to test the genotype of an organism. If the organism is homozygous in condition and we do experiment as test cross, then the offsprings, all the offspring are same. And if the heterozygous condition, if the genotype of an organism is heterozygous, then half of the offsprings will be homozygous and half of the offspring will be heterozygous. So let's talk about this question number 5. Test cross is an experiment. Test cross is an experiment in which the organisms showing dominance for a trait has to be tested for its genotype, whether the genotype is heterozygous or whether the organism is heterozygous or homozygous. This we have to test, and for this test we use homozygous recessive parent. So test cross is just an experiment in which the organism showing dominance for a particular trait has to be tested for its genotype, whether the genotype is heterozygous, whether the genotype is homozygous. What is the genotype of that organism? To test the genotype of that organism, we use this test cross as an experiment, and to test the genotype of an organism, we use recessive parent. Alright, so we use recessive parent. We use recessive parent to test the genotype of an organism. When any organism which is being tested for its genotype and showing dominance, it could be homozygous, or it could be heterozygous, Now let's talk about the test cross. We'll be going to design a test cross over here. As we know that to test the genotype of any organism, we will use recessive parent. We will use homozygous recessive parent. So let's talk about the height of a plant. If any organism is homozygous tall. Let's talk about the first case. We are going to discuss two cases. This is first case in which the organism is having, suppose the organism is having capital T condition, that means homozygous dominant condition, but we don't know what is the basic, what is the genotype of that organism. We have to still test the genotype of this organism. Now to test the genotype of this organism, we will mate with this organism is small t. We are going to mate this organism with recessive parent. This is the recessive parent, we know. This is the homozygous recessive parent, and in a test cross, which is an experiment to determine the genotype of an organism, always the homozygous recessive parent is being used. Now in case number 1, if any organism is homozygous dominant, then what we do in a test cross, we mate this organism with recessive parent. Now what is going to happen? It will produce gamete as capital T, it will produce gamete as small t. All the offsprings without exception in F1 generation will be tall. All the offsprings in F1 generation will be tall. That means heterozygous tall. All the offsprings will be heterozygous. So this is the conclusion, this is the result. We don't know the genotype of the parent of an organism or an organism. We are just testing the genotype of that organism, and for testing we are going to mate this organism with recessive parent. Now what happens? These two are going to make gametes. This has made capital T as gamete and this has made small t as gamete. When they fuses with each other, they produces offsprings of F1 generation. All the offsprings in F1 generation are heterozygous tall. All the offsprings are heterozygous. If in the conclusion of a test cross in the F1 generation we found the offsprings, all the offsprings are heterozygous in F1 generation, then we can say that the organism, by this conclusion we can say that the organism, what organism we are talking about? This organism. The organism is homozygous. The organism is homozygous. Alright. So by this conclusion we can say that the organism is homozygous in condition. Now let's talk about the second case. Suppose we don't know the genotype of the organism. Suppose we are taking the heterozygous condition here. We don't know the exact genotype. We will conclude a result from the F1 generation of it. Let's talk about here the next case, that is heterozygous condition. We will mate this with recessive parent again. We will make this possible. We will mate this organism with recessive parent. This will produce two types of gamete, as this is heterozygous in nature and this is going to produce only one type of gamete. Alright. Now what happens in F1 generation? Half of the progeny in F1 generation will be heterozygous and half of the progeny in F1 generation will be homozygous in condition. Alright. So what we will get by the conclusion from F1 generation in second case, that is half of the progeny is homozygous, half of the progeny is homozygous and half of it is heterozygous. Alright, half of it is heterozygous. So we can conclude now by making these organisms mating with recessive parent. By making these organisms mating with recessive parent, we can conclude the result if all the organism in F1 generation are heterozygous in nature, then we can say the organism is having homozygous condition, homozygous genotype. And if all the progeny, we observe the progeny in F1 generation, all the progeny in F1 generation, if found to be half are the heterozygous and half are the homozygous, then we can conclude that this organism is actually heterozygous in nature. So this is the complete solution of question number 5.